So the COVID primer puzzle is an interesting context which was devised by Tara Christie from the University of Sydney, and it relies on some stimulus material which is basically the design of primers that will test for COVID. And the idea is that these primers, shown in green, blue, orangey yellow and purple, they all amplify different sections of the COVID genome and will particularly amplify sections of particular mutants. So the primers will amplify the wild type, but only the purple forward primer here will amplify this particular variant, and the purple reverse will amplify this particular variant, and so on. There is actually another variant which is not shown to the students, but is something that they later have to work out. So each of those primer pairs is going to produce a different length of amplicon in a different variant of the COVID. So what the students are set in this particular instance is a puzzle where we have all of those primers. We've got the 4 forward primers, green, blue, yellow and purple, and 3 reverse primers, but our research assistant has mixed them all up. So we don't know which primer is which. We know that the forward primers are all forward and the reverse primers are all reverse, but we don't know in which order they are within that. So what we have to do is work that out. We're given obviously the qPCR buffer, and we're given some samples of cDNA for each of the variant types. So as an example of the way that you'd set this up, a student might generate a plate with 4 columns and 1 row, and into all of these wells they might pipette 5 microliters of each of the primers. Now clearly there's not going to be enough room for that, so we're just going to do primer number 1 into here. So let's put primer number 1 into each of these, and then we'll follow that up with a dispensing of all of the reverse primers, all 4 of the reverse primers, into each well. Now I should be changing the tip each time here, of course, to avoid the contamination, but we'll do that. Then we'll put in 5 microliters of wild type. Now again, we could use different combinations, and in fact the students will have to use different cDNAs to be able to identify every possible combination. And then we'll add 10 microliters of the buffer. And that will have set up the reactions. So there's a forward and reverse primer, there's the buffer, and there's the cDNA. So these are ready for qPCR, and we'll give them a little mix. And you'll notice that there's this plate button up here. Pressing that button effectively takes this set of 4 wells through an entire qPCR cycle with all the melts and amplifications and so on. And it gives all the amplification data. Now that looks a bit daunting, but if you just click on it and copy, and then go to Excel, and if we make a new worksheet and in there paste those values, you can see we've got the cycle run, the cycle of the run along the top. So these are each cycle, and then we've got the amount of amplicon in these wells here. Now, right at the end, we also have the length of whatever amplicons have been produced, and sometimes there may be more than one, and we've got the TMs that would result from the melt curve of that run. Now you can imagine there's an awful lot happening in the background here because the programmers had to work out exactly what cDNAs are in there, and I mean cDNAs, it could be more than one, and then what primers, and again, could be more than one of each. So it's all very authentic. If the students contaminate or if there is some mixing, ambiguity, whatever, you'll get more amplicons, more TMs, and you'll get melt curves that are commensurate with that. Now, what's quite fun is to plot out the melt curve. So select all of those values and then just insert a graph, and you can see there that 3 of the runs have amplified stuff and 1 of the runs hasn't. And that's a visual way of showing it. We could have just looked at the end, of course, and seen that we have these amplicons and these TMs for 3 of the runs that look as if a product has been made. So how big are those products? Where do they fit in? Well, one thing the students can do is look at the actual sequences, and there is a document that actually shows where the sequence alignment of each of these things. So this is where the primers are meant to bind. This is where the green ones are meant to bind, forward and reverse. So the student should be able to work out what sort of amplicon they're going to get from that. So although they can't really work it out quite so easily in terms of length from here, they can work it out from here. Now, I'm not sure whether Tara gives them this, but we do actually have a little bit of a cheat sheet for ourselves when we're going through this. And this tells us what amplicon size each forward and reverse primer combination will make for each species. So if we had a look at what we had achieved there, then what we would see is that the amplicons that we've managed to produce are 120, 87 and 144, and that looks like this, doesn't it? So that looks like the forward primer must be blue, and then the other ones are green, blue, red, reverse, and the one that didn't produce anything was magenta. So you can see that it is quite easy, if the student does everything correctly, to work out what has happened in these incubations, what amplicons have been produced, and therefore what the identity of F1, R1, R2, R3, and R4 must be. Now, of course, as soon as they've done that, they're one step ahead in working out the other ones, because as soon as you know what the identity of these are, it makes it quite easy then to set up incubations that would tell you what the other ones are as well. But what most students end up doing is simply creating a 4x4 grid, 4 rows, 4 columns, and doing all the forward primers and all the reverse primers in a sort of matrix. And then they plot all of those results. So they can do it all very, very quickly. So it's a great little puzzle. It involves them doing this part of it correctly, the pipetting part. There's organization, there's design, and there's execution. You've got to have results that you rely on. And then there's the actual processing of those results and marrying them up with the different possible combinations of forward and reverse primers that will produce particular amplicons in particular variants. So a very, very good assignment. And the other thing, of course, is that the data is always being sent to the database. So the combinations that we've just used in this session, RGHM-TZ, they are, this RGH-TMZ1V, they have been recorded in the database. So the instructor knows what F1, F2, F3, F4 were, and similarly with R1, R2. So, and every student is going to get a different combination, and it leads to a totally non-plagiarizable assignment where everybody's got the same task, but the results are different. And even the logical steps that someone might go through to nut it all out are going to be slightly different because they're getting slightly different information. But the strategic approach is always the same. So that's a really neat assignment. We do have really nice exemplars of amazing reports that students have done for those that are interested.